My name is Uilin and my metric number is 206904. Today I will conduct an experiment on alcoholic fermentation in yeast. The materials used in this experiment are empty plastic bottles, warm water, yeast, starch, glucose, table sugar, and balloons. First, label all the empty bottles 1 to 4. Bottles 1 to 3 will have yeast, carbohydrates, and water. Bottles 4 will have only yeast and water with no carbohydrates. Then, fill the bottles with 30 ml of warm water. Add 9 teaspoons of carbohydrates into bottles 1, 2, and 3. Do not add carbohydrates into bottle 4. This will be the control of this experiment. Then, add 3 spatula of dry yeast into each bottle. Then, mix it with gentle shaking. Cover the opening of each bottle with a balloon to seal the ends of the bottles. Observe the bottles and record the observations in the table. Then, every 5 minutes for 25 minutes, observe what occurs in the bottles and any changes in the balloons which cover each bottle and record the observations. The carbohydrates and yeast in bottle 1, 2, and 3 cause foaming due to the fermentation. In this experiment, we tested the ability of yeast to ferment different carbohydrates. The carbohydrate glucose is a monosaccharide or simple sugar. The other carbohydrate sucrose is a disaccharide which is made up of 2 simple sugars. Sucrose is composed of link, glucose, and fructose monosaccharides. The carbohydrate starch is a polysaccharide which composed of monosaccharide units bound together by glycosidic linkage. Only monosaccharides can be used directly by yeast in fermentation. This means that sucrose and starch must be broken down to their component monosaccharides by the yeast before fermentation can occur. As carbon dioxide is produced, carbon dioxide covers the space in the bottles and cause the balloons to inflate. Alcoholic fermentation is a biochemical process in which sugars are converted into small amount of ATP, producing ethanol and carbon dioxide during the process. Alcoholic fermentation is the main process that yeast cells the anaerobic organisms used to produce ATP. In the absence of oxygens, alcoholic fermentation occurs in the cytosol of yeast. Alcoholic fermentation begins with the breakdown of sugars by yeast to form pyruvate molecules, which is also known as glycolysis. The two molecules of pyruvate are then reduced to two molecules of ethanol and carbon dioxide. Under anaerobic conditions, the pyruvate can be transformed to ethanol, where it first converts into a midway molecule called acetaldehyde, which further releases carbon dioxide, and acetaldehyde converts into ethanol. In alcoholic fermentation, the electron acceptor called NAD+ is reduced to form NADH. The exchange of electrons that occurs in the process helps to build ATP.